Good morning, dear students. I am Dr. Anmol Kumar, Assistant Professor, Department of Biotechnology, Atme University. Today I will take the topic of preparation of sections, staining and mounting of slides. In my previous video lecture, I have discussed about the various types of microtome, and I also discussed about the different steps of the tissue processing. So in the tissue processing, you have processed the tissue and finally made the block. Now that block is ready for the sectioning, and finally you will get the section. So in this video I will discuss how to get the section from the microtome and finally how to do the staining process. So these are the major equipment required for the sectioning of the tissue block. So first of all you need microtome, then glass slides, then hot plates and water bath and ice tray and hot air oven. These are the minimum requirement for the sectioning of the tissue. Other than this one, you also need the forceps, brushes and pencil needles and adhesive. Those adhesive will be used for the adherence of the sections on the glass slides. So if all the instruments are ready, then you can go ahead for the sectioning of the tissue. Now I will discuss about the paraffin sectioning procedure. So I will give some outline of this procedure. So first of all you should trim the tissue block, because while doing the embedding process the wax material get deposited on the tissue block. So you should remove this unwanted wax material. And then after removing the unwanted wax material you should give some proper shape like triangle shape, trapezoid shape, rectangle shape, right? After giving the rectangle shape then or triangle, whatever it may be, it doesn't matter, then you transfer the ice to tissue block to the ice, right? In the ice what will happen, it will absorb some amount of moisture which helps during the sectioning process, right? After the chilling of the tissue block, then you fix the tissue block into the sample holder of the microtome, right? In the meantime you also fix the knife into the blade holder of the microtome, and you reset the thickness gauge around 4 to 5 micron, because 4 to 5 micron is most commonly used for the most of the sectioning process. So this is all about the procedure. Now we will start the actual sectioning process in the next slide. And you know that if you turn the one complete wheel you get the one section. So when everything is ready, so you should also ensure that the tissue block, the surface of the tissue block should be parallel to the knife, so that you will get the serial of the sections in the form of ribbon. So each length is around 10 to 12 inches, right? So if you will get this type of ribbon, then you carefully transfer onto the water bath. So in the water bath you have to set the temperature of the 40 degree. At the 40 degree what will happen, the section of the tissue whatever you are getting after the microtomy, it will become flat and it will remove the wrinkles while generating during the microtomy. So you can see in the picture, in the first picture you can see how to get the sections using the microtome, and after getting the sections the ribbons of the have been transferred onto the water bath, and finally your sections become flat here. Then after transferring to the water bath you need to transfer finally onto the glass slide. So carefully you take the glass slides. The glass slide should be labeled and it is charged. Charge means it will help to the adherence of the sections. So you submerge the slides onto the water and carefully take this section onto the glass slide, and finally you drain the water by keeping in upright position, and you transfer the glass slides to the hot air oven. So what will happen in the hot air oven, it will melt some wax, so it will help the adherence of the section to the glass slides, so it will easy and during the staining process, and it will not come out while washing during the staining process. Now your slide is ready for the staining and you can go ahead for the staining process. So these are some sequence of events during the section preparation. I mentioned here first pictures you can see here, this is the block trimming, then microtomy, and now the ribbon of the paraffin section ready, and you should transfer onto the water bath, and finally the ribbon will be transferred onto the glass slides. This is all about the section preparation. And then this is the staining process. This is the last part of the histopathological process. So I will explain this staining process in the next unit of this paper. So here I am giving the outline of the staining process, so you will get the complete idea of the histopathological process here. So staining is actually enhancing the contrast of some particle detail of a tissue, right? A particle detail in the sense you can contrast the elements or some microbes, even you can expose some antigens. So it will help in the diagnosis of any disease in the tissue section at the microscopy level. This is the your goal of the staining. So there are several types of stainings are there. So I am explaining here only the hematoxylin and eosin staining. This is the most commonly used staining in the lab. This is also called the routine staining process. So hematoxylin is usually stain the nucleus, so while the eosin will stain the cytoplasm. So before the staining process, you have to prepare the slides here. So this is called the pre-staining process. So you need to remove the wax, because all the staining process need to be done in the water soluble staining solution. So you have to remove the wax and you have to hydrate the tissue. So in this case, first of all, you have to melt the wax at the heating block. So it will melt. After melting, you have to deparaffinage the glass slides in the xylene solution. So you have to repeat the process. Then finally all the wax will be removed. Then you have to rehydrate the tissue with the decreasing concentration of alcohol, and finally you have to put the slides in the pure water. Now your slides is fully hydrated and all the wax and xylene has been removed. Now it is ready for the staining. So here I just mentioned some key steps the hematoxylin and eosin staining. So first of all, you have to stain the tissue in the hematoxylin. So it will stain the whole tissue here, along with the nucleus. It will also stain the cytoplasm. Then after the staining, again you have to wash with water. See, after washing with water, you have to differentiate the tissue. Differentiate means you have to expose the nucleus. So it will only expose the nucleus part. Then again you have to wash with running water. Then you have to do the bluing step. In bluing step, what will happen? It will enhance the contrast of the nucleus and you will get a very good color of the nucleus. Then you have to counter stain with the eosin solution, and finally final wash with the running water. Now again you have to do the dehydration step here, because you have to remove the water, and because finally in the last stage of the staining process is mounting. In the mounting you have to add the organic solvent. So you need to remove the water in this case. So you have to dehydrate the tissue with the increasing concentration of alcohol. Then finally xylene, it will help in the clearing of the tissue. Now the last step of this staining is the mounting. Mounting is just a preparation of the tissue for the microscopy purpose. So you have to put some mountant like DPX on the tissue section and let it spread. After that you have to keep the cover slip in a tilted position and allow to fall gently onto the section. So it will allow the mountain to spread over the section and just below the cover slip, and let it dry for overnight, and next morning it is ready for the microscopy. So other than DPX, terpene and Canada balsam is also used for the mounting purpose. Now this is the final picture you will get in the microscope, and this is the picture of the hematoxylin eosin stain. See here the hematoxylin stain the nucleus while the eosin stain the cytoplasm part, right? And now you will diagnose the any disease based on the staining process, right? So that's all about the hematoxylin eosin staining. Thank you for this video. Hope you understand this lecture, and we will meet in the next lecture, which will be the last lecture of this topic. It is automated tissue processor. Thank you.